Hello everyone. Today we'll discuss the process of gastrulation in frog. In last class, we have discussed the different morphogenic movement during the process of gastrulation in frog, that are the involution, invagination, then migration, epiboly, emboli, all and the convergent extension. All these processes occur one by one or in a synchronized way to convert the blastula into the gastrula. What exactly occurs during the gastrulation? During gastrulation, the three germinal layers, namely ectoderm, mesoderm, and endoderm, are formed. The dorsal blastopore leaf. Here there is a start of inward movement just below the gray crescent area, and there is a formation of this lip is formed, and that is the dorsal blastopore lip. Along with that, due to the inward movement, there is a formation of the lateral and the ventral lips are also there. At the same time, the yolk plug is formed due to the epiboly or during the epiboly, and there is a start of invagination or inward movement of the cell and from the gray crescent area, and then the other processes, that is ingression and other processes like the migration, and all these processes continues. Then blastocele, or this is as it is formed from the blastula having the cavity, that is the blastocele, and these blastocele eventually, or due to the migration of the cell, inward movement of the cell, there is a formation of the another cavity, and that cavity is called as a Archenteron cavity, and this previous cavity, that is the blastocele, is displaced, and at the end this blastocele is entirely replaced by the another cavity, and that cavity we called it as a gastrocele cavity. So during the gastrulation process, it starts from the blastula. At the start of this, there is the inward movement of the dorsal cells from the dorsal blastopore, or from the area that we called it as a blastopore. Then this is the movement start continues, and there is a formation of the dorsal blastopore leaf. Along with that, there is a formation of the lateral and the ventral blastopore leaf, and there is a formation of the yolk plug. And after that, this the inward movement, and the what that is the epiboly movement, involution, invagination, all these migration, then convergent extension, all these are responsible for the formation of the three germinal layers. Outer one is called as ectoderm, middle one it is called as that is a mesoderm, and this inner one that is called as a endoderm layer. Means at the end of gastrulation, the three germ layers are formed. This is the same process, or the picture showing the process of that gastrulation in the frog. This is a laced blastula having this blastocele. This is an animal pole. This is a vegetal pole having the megameres, and this is a dorsal lip of the blastopore from where the inward movement of the cells will occur. This is the early gastrular stage. As the process advances, and in the middle gastrulation stage, the cells move inward, and so that this blastocele, it is now displaced by the another cavity, and this cavity is called as the Archenteron cavity. And this Archenteron cavity completely uh, take over the uh, completely replaces the cavity that we called it as a blastocele, and there is a formation of the three germinal layers. One is ectoderm, mesoderm, and this is the mesoderm, and this is the endodermal layer. So these are the steps or these are the processes involved in this process of gastrulation. The gastrulation begins when cell in the region of the gray crescent. This is the gray crescent area just below the equator. The cells from the gray crescent area move inward, forming a dorsal lip of the future blastopore. Future blastopore means this is the opening through which this will be the opening, and from where these cells, these are the elongated cells like the bottle cells that we call it as a bottle. This is the vegetal pole and animal pole, from where the cells will invaginate or the inward movement of the cell. As the movement of the cell advances, these are the bottle cells, and this is a what the tube-like structure, that is the Archenteron, starts to form. This is the cavity that we called it as Archenteron, and this is the previous cavity, that is the blastocele. Slowly, what will happen? This blastocele will get diminished, or this cavity or this Archenteron cavity will replace this blastocele. Okay. As the process advances, what happens? There is a formation of the dorsal blastopore lip, and as there is a inward movement of the cells occurs, what happens? This is the first one, is a what? Inward movement of the cell or the local buckle, that is a inward buckling of the cells, that is a invagination. Then these cells or these cells which are inward movement from the gray crescent area, they move along the previous sheet of the cells, and that is called as a involution. As this inward movement of the cells occurs, what happens? This is the dorsal lip of the blastopore. The another lip, that is a what? The ventral lip of the blastopore is also formed, and the lateral lip of the blastopore is also formed. And thus this is a yolk plug, and from this there is a what? Inward movement of the cell, that is what we called it as a what? Epiboly movement. So the cells from the animal pole spread out, pushing the surface cell below them toward across the dorsal lip, and these cells move into the interior of the embryo, where they form the endoderm and the what? Mesoderm. The Archenteron expands. This is an expansion of this Archenteron cavity. Later on, what happens? These are the deuterostome. This will become a what? The anus. This opening or the dorsal lip of the blastopore, or this is the dorsal lip of the blastopore, and this opening, that we call it as a blastopore. This blastopore, during the development, either it becomes a what? Mouth or anus. In protostomes, it becomes a mouth, and in deuterostomes, it becomes the anus. Frog is included under the deuterostome, and therefore this opening will what? That is a develop into the what? That is a anus region or the anal opening. The Archenteron expand, destroying the previous cavity that we called it as a this blastocele. Then the blastopore lip forms the circle, with the cells moving to the interior around the blastopore. The yolk plug is visible and through this blastopore. So these are the steps how the what? Three germinal layers are formed during the process of the what? The gastrulation in the frog. This is the how the formation. This is the dorsal lip. It is formed, then this dorsal lip expands, and after that there is a formation of the what? The lateral lip, and later on the what? The ventral lip is formed, which completes the circle. And due to this is the formation of the yolk plug. And through this yolk plug, the cells will migrate toward the inward position. These are the what? How there is a formation of the opening, that is called as a blastopore. The what are the steps during the what? The gastrulation process. This is the what? The blastula. This is the blastocele cavity. These are the larger cells. These are the megameres. These are the micromeres. This is the animal pole, and this is the vegetal pole. This is the equatorial region, and below equatorial region there is a gray crescent area. These are the superficial cells or the outer cells. What happens? The cells which are in the gray crescent area, they starts to form there is a formation of the dorsal blastopore lip. This is a dorsal blastopore lip from where the local inward buckling of the cell, that we called it as the invagination. The shape of these cells, it get changes and these cells become a bottle shaped cells, and these are called as a bottle shaped cells. And this is the first, how there is a what, the first step, that is a blastopore formation, or the formation of the what, that is a the opening, and this blastopore is going to form the anus in frog, as frog is included under the deuterostome. The blastopore is the first opening in the embryo, the point of invagination during the gastrulation. The blastopore is eventually become either mouth or anus. In frog, it becomes a anal opening, one end of the gut tube or the other. The space that forms during this time is the primitive gut or Archenteron, means the tube which will get formed during the what, the process. This is the tube, or this is the tube. This is the cavity, that is the Archenteron cavity, and this is the tube, and this will be the anus, and this will going to form the digestive tract, or that is we called it as a gut. The frog blastula is many cell layer thick. The cells of dorsal lip originate in the gray crescent, just below the equatorial part, and invaginate to create the Archenteron cavity, or it is called as a gastrocele. The cells continue to move from embryo surface into the embryo by the process that is the involution. Means, first of all, the movement of the cell which occur, that is called as the invagination. And after that, these cells continued, and another movement of the cell occur, and that we called it as a involution. And these cells become the cells which are involved in the involution and the invagination. They form the endoderm and the mesodermal layers. Then the invagination process. In frog embryos, gastrulation is initiated at the future dorsal side of the embryo, just below the equator in the gray crescent area. Here the marginal endodermal cell sinks into the embryo or shows the inward movement, and thus forming an opening, and that opening we called it as a blastopore. And these cells now change their shape, and these become a flag shape. So see, these are the marginal cells, and these marginal cells starts to move inward direction, and so that there is a formation of the opening, and this opening we called it as a blastopore, and this cavity, that cavity we called it as a Archenteron. This is the what, the structure of the electron micrograph picture of the blastopore during the invagination, the inward movement of the cell. So these cells changes their shape, they become elongated, and it having the neck, and these therefore these are looking like a bottle, and therefore these cells are called as a bottle cells. These are the bottle cells, and that maintain the contact with the outer surface with the cytoplasmic strands, where their main body is displaced toward the inside of the embryo. Therefore, in frog, gastrulation begins in the marginal zone below the equator of the blastula, and where in the gray crescent region. Here the endodermal cells are not so large or so yolky as the most vegetal blastomia. Thus, although the bottle cells may be responsible for initial growth, the motivating force, this appears to come from the deep layer of the marginal cells. Furthermore, this deep layer of the cells appear to be responsible for continued migration of the cells in the embryo. This is the process of invagination. See, you can observe that there is a, due to the invagination or the inward movement of the cell, there is a formation of the opening, that is a blastopore, and this is a cavity, that is a Archenteron. This is a what? another cavity. This is the blastocele cavity. Slowly what will happen, this Archenteron, it will enlarge and it will form the gut or the digestive system of the embryo. This blastopore, it will form the anus of the embryo, and this slowly what will happen, these blastocele will get displaced by this cavity or by this Archenteron cavity. The apical constriction of the bottle cell drives the blastopore and the first movement of the cell, that is the invagination. This is the same photograph, that is what this is the word, the blastocele cavity, and this is the blastopore. This is the dorsal blastopore lip. Then the lateral lip will form, and then the ventral lip will form, and so that here there will be the formation of the word, the plug or the yolk plug, through which the cells will move inward direction. And these are the long cells, these are the bottle cells. This, first of all, this is the invagination, and then these cells will move along the marginal cells or the just lying adjacent to the marginal cells. The process, it is called as a involution. See, this is the site of the Blastopore Formation, from where just below the equator at the dorsal side, and from where the what, that is the inward movement of the cells. These are the marginal cells, which are the endodermal cells. They are moving inward direction, and that is called as the invagination. And after that, what happens? These cells, after inward movement, they are moving along the these previous layer, that is, these cells are moving along this layer adjacent to this previous layer, and therefore the moment we called it as a involution, and these are the bottle cells. So these bottle cells, they are moving, and this is the blastopore, that is the opening which will going to form the anus, and this is the digestive tube. This is the Archenteron cavity, and this tube like structure which will going to form is a digestive tract or the digestive tube. Then this is the what inward movement of the cell, or the showing the, this is a blastopore leaf, and this blastopore leaf, it what this will be formed. This is the inward movement, this is the Archenteron formation, and now these cells, after the formation of these, the whatever the cells are migrating, they are this is the bottle cells, and whatever the cells are migrating, or these are spreading along the previous layer, and that is called as a what, the involution movement. These are the notochord precursor cells, and this is the blastopore leaf. Then the second process, that is called as a involution. As just we have discussed, the what happens as the inward movement of the cells occur, that is the this inward movement of the cell occur. These are the what? These are the bottle cells, and these again changes their shape, and they move along adjacent to the what, this previous layer. And as these cells or this zone, it moves along the previous layer, what will happen? This blastocele will slowly get replaced by the cavity, and this cavity is called as a what? This cavity is called as a Archenteron cavity, and this involution occurs around the dorsal lip of the blastopore. And these are the marginal cells. They are moving in inward direction, and the second step during the gastrulation is the involution of the marginal zone cells. So the next phase of gastrulation involves the involution of the marginal zone cells, while the animal cells undergo epiboly and converge at the blastopore. Why? Because at this time there is a now formation of the dorsal lip, lateral lip, and the ventral lip. On reaching the tip of the blastopore, the marginal cells turn inward and travel along the inner surface of the outer cells of the shapes. The cell constituting the lip of blastopore are constantly changing. The first cells to form the dorsal lip are the endodermal cells that invaginated to form the leading edge or the leading part of the Archenteron. And these cells, which are first of all invaginated, they will become the pharyngeal cells of the foregut. As these first cells pass into the interior of the embryo, the blastopore lip composed of involuting cells that are precursor of the head mesoderm. Then the next cell involuting over the dorsal lip of blastopore or the called the mesoderm cells. Means first of all the cells this moved inward. These are the pharyngeal cells of foregut. Then the cells which move inward or involute, these are the cells from the head mesoderm. Then next, these are the cells from the chorda mesoderm. And these cells will form the notochord, a transient mesodermal backbone, and that is essential for initiating and differentiation of the nervous system. Then the third process, when the what these cells have started moving, what happens? The next is the convergent extension. We have discussed the what is the convergent extension. This if the four cells are arranged in a four tiers, what they become elongated. They become in a two tiers. If the what the cells are present in the what in one single row, if the cells are present that is in 15, then 15, then in next line or next row 15, then next in another row 15, and the miss if we are having the what that is a four layer of the 15 cells each, then during the convergent extension we will have the two rows each having the 30 cells, and that is the what extension or stretching. And so that what during after the what the first process is the inward movement, that is a in a inward movement, and then that is a in that is a ingression, and the what that is a in then the what in involution and the ingression, and the third process that is a convergent extension. What is the convergent extension? The dorsal mesodermal layer cells which goes inside जो migrate हो रही है और which cells which are migrating inside, these cells are going to form the dorsal mesoderm, and it spreads over the entire embryo, and these cells replaces the blastocele cavity. These red color cells undergoing the extension, and so that they are replacing, this is the dorsal mesoderm which is extension, that is a undergoes the convergent extension, and so it replaces entire cavity from here, that is a this blastocele cavity is replaced, and the what as the inward movement advances, the another cavity that is the Archenteron progresses. The next after the convergent extension, the epiboly. As the new cell enters the embryo, the blastocele is displaced to the side opposite to the blastopore leaf. See, during the epiboly, this diagram we have already discussed. During the epiboly, what happens? This yolk plug is formed. As first of all, there is a formation of the dorsal lip. Then there is a formation of the lateral lips, and lastly the what ventral lip, and therefore there is a formation of the what yolk plug, and this is the what the blastopore lip. So how the epiboly, the as the new cells enter the embryo, the blastocele is displaced to the side opposite to the dorsal blastopore lip. Meanwhile, the blastopore is displaced. Vegetal pole widens as more animal hemisphere cells converge at the blastopore. The widening blastopore develops lateral lips and finally the ventral lip. Means during the epiboly what happens? Simply the inward movement of the cells causing formation of the first of all there is a dorsal blastopore lip. Then the widening of the blastopore, it develops the lateral lip as well as the ventral lip over which the what the additional mesoderm and endodermal precursor cell pass. With the formation of the ventral lip, the blastopore has formed the ring around large endodermal cell that remain exposed to surface, and that we called it as a yolk plug. Means during the epiboly, what happens? The dorsal blastopore lip is there. There is a formation of the lateral blastopore lips, and the later on there is a formation of the ventral, and this completes the entire circle. And so that what happens, the blastopore form a ring around a large endodermal cell that remain exposed on the surface. The remaining patch of endoderm is called yolk plug, and it too is eventually internalized. At this point, all the endodermal precursors have been brought into the interior of the embryo. Means when there is a formation of the yolk plug, all the endodermal cells have moved to the inward side, and ectoderm has encircled the surface and mesoderm has been brought between them. Means when there is a epiboly, what happened? All the endodermal cells have already moved inside. Then in the middle, there is a mesodermal cells, and at the outer portion, the what? The ectodermal cells are there. That is the means. It is the step where the what the three germinal layers are more or less take the what that is their position, that is the ectoderm at the outer side, then the mesoderm at the middle side, and the endoderm at the what the inner side. This is the epiboly of the what that is a ectoderm. That is a ectodermal layer flattens and it surrounds the entire embryo, and this is the movement that is called as a epibolus movement. So this is the epiboly of the ectoderm. It moves the what that is a this ectodermal layer. It surrounds the entire embryo. All the endodermal cells have already reached at the inner position, and the middle part there is it is occupied by the mesodermal cells. And this is the epiboly of the ectoderm, and this the formation of the yolk plug by the what that is as first of all there is a dorsal lip, then the what that is a lateral lips are formed, and then the ventral lip, and this forms the circle. Those cells which are not moved, that as called as a yolk plug, and by this movement all the cells of the endoderm have moved inside the embryo. And the last phase during the gastrulation is a mesenchyme migration. This is the mesenchyme migration, or these middle layer cells. They are migrating within a what? The coelomic cavity, and they are in the middle. The outer layer, it is occupied by the what? That is by the what? Ectoderm layer. And this is a endoderm, that is a endodermal layer, that is a yellow colored cells. These are the endodermal cells. This is the cavity that is called as a what? The Archenteron cavity that will later get formed and develop into the gut or digestive system. This is the opening that is called as a blastopore that will develop into the what that is a anus of the what that is a the frog, and the first organ which is going to form, that is the gut in the embryo, and three germinal layers. Outer one is called as a ectoderm. This is the middle layer, that is a the mesoderm, and inner layer, that is the inner layer, that is the endoderm. This is the mesenchyme migration. This occurs by the what that is a migration process, or the migration, the cell separates and they are migrating to their way, and they are forming the what that is a mesoderm. Here at the ventral side, this is the what the ventral mesoderm is formed, and these are the what that is a mesodermal layer, and which occupies the central position or the middle position. And these outer blue color cell, these are the ectodermal, and the yellow colored cell, these are the endodermal, and the red color cell, these are the what the mesodermal cells. So this is the last movement, that is a mesenchyme migration. So this completes the what that is a entire process of the gastrulation. And so at the end of the gastrulation, the three germinal layers are formed. There is a what the blastopore opening that will result in the formation of the that is the anus. This cavity is going to form the, this is the Archenteron cavity. This is going to form the what, the gut or digestive system. Here is the what, the yolk plug, or the what, that is a yolk plug, and there is a formation of the what, this outer layer that is called as a ectoderm. This is the mesenchyme or the mesodermal layer, that is the mesenchyme migration, and the yellow colored cells, these are the mesodermal cells. And so the at the last there is no blastocele. Blastocele is entirely replaced by the gastrocele or archenteron cavity, and the three germinal layers are formed. So this is a complete process of gastrulation in the frog. So this is the ectoderm. Then this ectoderm is going to form the what, skin and the central nervous system, brain, spinal cord, neural tube, then bone, cartilage, connective tissues in the head. Then this is the middle layer, that is a mesodermal layer, and this middle layer, it is going to form the blood vessels, notochord, bones, cartilage, circulatory system, and the urinogenital system. Then next to that, the endodermal layer. That endodermal layer, that is the inner layer, and this inner layer, that is first of all, it will form the gut along with the what, the vital organs like the liver, lungs, pancreas, and the salivary glands. And later on, there is a formation of the neurulation, and the neural tube will form, and the nervous system will develop. So this is the this these cells, these are the endodermal cells, these are forming the gut. These pink color cells, these are the mesodermal cells, these are forming the notochord, and the neural tube or the nervous system, it is formed by the outer layers of the cell, that is a ectodermal cells. This is the what, how the that is for the what, the how it is there. So thank you.